In this final chapter, we will be discussing genetic engineering. In the process, we will learn how we can benefit from genetically engineered products, what we have learned from this, how does genetic engineering work. In an attempt to find a better and easier way to grow corn, scientists have genetically modified it. This is true of soybeans, tomatoes, potatoes, cotton, and many other crops. But we are going to focus on corn for now. It is the second most consumed crop right after wheat. We've eaten it plain, corn syrup, cornmeal, or as corn starch. We also eat it indirectly by eating the livestock that is grazed upon it. If you were to actually think about what was happening in the cornfield, you will notice a very small ecosystem, one of cooperation and competition. The corn stalks are happy because they get air, sun, and nutrients, but then the weeds start to move in and try to steal the nutrients from the soil. And what about the insects? Some insects try to feed off the corn. The farmers have to do a lot of work in order to cultivate it. He or she has to manage this ecosystem so that the corn can thrive. This takes time, human energy, fossil fuels for the tractors, and chemicals for protecting it. But how much of this is doing more harm than good? We now have genetically modified corn that can produce its own pesticides and resist herbicides. This means there's less dangerous chemicals being used. So how do we make the corn create these chemicals? Each cell in the corn plant has a complete set of instructions on how to grow and reproduce. This information passes from one generation to the next and is called the genome. Genes are short sections of instructions that govern specific reactions, chemicals, and events in the cell. If a gene is changed, then the inheritable trait changes, such as making corn produce a new chemical such as an insecticide. A soil bacterium has the genes to make insecticidal proteins. So by taking the gene out of the bacteria and inserting it into the corn plant, we have corn plants that produce an insecticidal protein. Like the corn plant, we have our own chemical database of information. We also have genes and genomes. All genetic information is stored in the nucleus of the millions of cells in our body. Each nucleus contains chromosomes, which are 46 compact structures of intertwined molecules of DNA, and about 30,000 gene components that convey one or more hereditary traits. DNA is a special template written in a molecular code on a tightly coiled thread that carries all the genetic information. Let's take a closer look at DNA. So what exactly is DNA? DNA is made of a fundamental chemical units repeating over and over again, like a polymer we saw in the previous chapters. Each unit is composed of 3 parts: a nitrogen-containing base, the sugar deoxyribose, and a phosphate group. There are 4 different bases which these units are made of. They're adenine, guanine, cysteine, and thymine. A nucleotide is a combination of a base, phosphate group, and deoxyribose. This nucleotide is an adenosine phosphate. Any of the 4 bases, which are right here, could have been used to form this nucleotide. By changing the structure of the base, you can change the nucleotide. The other 2 parts stay the same, the deoxyribose and the phosphate group. There are two covalent bonds holding the nucleotide together. Remember, a covalent bond is when two atoms are sharing their electrons. One bond exists with the phosphate group in the sugar. The other bond is present between the nitrogen of the base and the ring carbon of the sugar. When the nucleotides are linked up, it is when you get the DNA structure. So what does that look like? A typical DNA molecule consists of thousands of nucleotides covalently bonded in a long chain. The phosphate groups are responsible for linking each nucleotide. So the pictures in yellow here, you can see that that one oxygen of the phosphate group is holding on to one nucleotide, while another oxygen in the same group is holding on to a different nucleotide. Notice the OH group that has been circled. This OH group is where the phosphate attaches. A phosphate group of one nucleotide reacts with the OH group on the deoxyribose ring of another nucleotide, forming and eliminating water. The total DNA structures consist of thousands of nucleotides all linked up. You may wonder how the DNA structure was ever determined. Rosalind Franklin used a technique called X-ray diffraction. When a crystal is hit with a beam of X-rays, it generates a pattern that reveals the position of atoms in the crystal. This technique uses the fact that molecules' electrons diffract X-ray rays at particular angles and resulting in a pattern, like the one you see here. And can be used to solve the structure of a crystal. Watson and Crick saw this pattern of the DNA molecule and combined it with the work they were doing. They were then able to put the pieces of the puzzle together to build a model of the structure. Using Rosalind Franklin's X-ray diffraction data, Watson and Crick proposed a molecular model for DNA. This model had a double-stranded of repeating nucleotides, which is the structure we saw earlier. This would be one nucleotide. The two strands are lined up with complementary bases. The thymine is always paired with the adenine. Notice how the A and the T are always together. And the cysteine is always paired with the guanine of neighboring. Notice that the G and the C are always together. These are held together by hydrogen bonds between the two, which are the red dots. Remember, hydrogen bonds are not real bonds, they are attractions. So the strands are being held together by attractions, not by bonds. The nature of the base pairing required that the two strands be coiled in the shape of a double helix. As mentioned earlier, adenine hydrogen bonds with thymine and cysteine with guanine in DNA. Notice that the red dots indicate that it's just an attraction, not a true bond. The real bonds are the black solid lines. But this attraction is strong enough to keep the two strands together. Erwin Chargraff's research showed that for all humans, the percentage of adenine is almost identical to the percentage of thymine. Similarly, the percentages of guanine is almost identical to those of cysteine. From this, Chargraff concluded that the bases always come in pairs. Adenine is always associated with thymine, and guanine is always associated with cysteine. DNA exists in every cell of our body. So how does it get there, and how is it always the same? The process by which copies DNA are made is called replication. The original double helix partially unwinds to make two complementary portions separate. So notice how it kind of unzips here, and it has two different portions down here. Each of the strands serves as a template for the synthesis of the complementary strand. So the new strand attaches to the original, and because C is always with G and T is always with A, they know where to line up. The result is two complete and identical DNA molecules. A complete set of genetic information is packaged into chromosomes, which is packed into the cell nucleus. Chromosomes are rod-shaped compact coils of DNA and specialized proteins packed in the nucleus of cells. As the nucleus splits into daughter cells, each chromosome is rapidly duplicated. 3 billion base pairs in each human cell provide the blueprint for producing a human being. The specific sequence of base pairing is important in conveying the mechanism of how genetic information is expressed. The expression is seen through proteins. Through directing the synthesis of proteins, DNA can control the characteristics of an individual, including inherited illnesses. Proteins are made of amino acids. We talked about that in previous chapters, but let's have a little review. The general formula of the amino acid consists of a carbon with 4 groups coming off of it: a carboxylic acid, an amine group, and a hydrogen. And finally, on the 4th, we have the generic R. Remember that an R group is a generic representation of any organic chain. There are 20 naturally occurring amino acids that make up proteins. So there are actually 20 different R groups that can be substituted right there. Two amino acids can link together via a peptide bond. The two molecules join, expelling a molecule of water, then the peptide bond is formed. Notice how there's an OH on the carboxylic acid and an H on the amine group. These two come together to make water, and then that nitrogen and the carbon that they were bonded to come together to make the peptide bond. This process may repeat itself over and over, creating a peptide chain. When many of these are connected, the protein is formed. Once incorporated into the peptide chain, the amino acids are known as amino acid residues. These are amino acids that have lost the hydrogen in the OH group in the process of bonding together. The order of bases in DNA determines the order of amino acids in a protein. Because there are 20 amino acids present in the proteins, the DNA code must contain 20 code words, so to speak. Each word represents a different amino acid. The genetic code is written in groupings of 3 DNA bases called codons. This diagram shows the codons, determined according to the base sequence of the nucleic acid strand. The expression of the genetic information is seen through the specific proteins assigned. So you notice how they have 3 letters, and these 3 letters will relate to a specific protein. We have discussed proteins in previous chapters. We have also talked about them in our food, and we've talked about them in polymers. Now we're going to take a closer look at the structure of proteins. In the previous slide, I mentioned that the individual amino acids were linked together through peptide bonds. This sequence of amino acids is called the primary structure. We can tell a little bit about the protein by knowing the primary structure. We know what it's made of and how it will react in certain environments, but it's not until we look at the larger structure of the protein that we know the larger functions. After the primary structure has been all linked up, one part of the protein strand will be attracted to other parts of the protein strand. This has to do with the attraction of atoms. If there's an extremely electronegative atom close to a hydrogen bonded to another electronegative atom, the hydrogen bonds will occur. Once again, the hydrogen bonds are attractions, not real bonds. So if we look here, we have oxygen and we have a hydrogen bonded to a nitrogen. Well, the oxygen and the hydrogen here are attracted to each other, and there's your hydrogen bond. The first time this happens, the secondary structure is formed. The secondary structure of a protein is the folding pattern within the segment of the protein chain. Alpha helix and beta pleated sheet are the two most common secondary structures. Notice that with both pictures, there's dotted lines signifying the hydrogen bonds. These are the attraction of one atom to the neighboring hydrogen. This is what's causing the structure to be formed. Once the secondary structure occurs, we zoom out a little bit more and look at the whole protein. There are similar interactions throughout the structure, which then determines the shape of the protein. This overall molecular shape of the protein defined by the interactions between amino acids far away from each other in sequence. However, they're close to each other when they're coiled up or overlap. The sequence is characterized by the amino acid, which is called the N-terminal at the end, and the carboxyl terminal, or C-terminal, at the other. Since the amino acids are linked up in the same way, it always starts with the nitrogen and ends with the carbon. So even though it continues on, it will always be nitrogen first and then carbon. The function of protein is dependent on its shape of three-dimensional structure. Small changes in the primary structure can have dramatic effects in its properties. Sickle cell anemia is an example of a condition that develops when red blood cells take on distorted shapes due to an error in the amino acid sequence. Because these cells lose their normal shape, they cannot pass through openings in the spleen and other organs. Some of the sickle cells are destroyed and anemia results. Other sickle cells can clog organs so badly that the blood supply to them is reduced. So let's turn our attention back to our original discussion of corn. How do we go from an original species to modify it to be more resilient, to produce better tasting food, and so on? This is the process called genetic engineering. When a species is genetically engineered, the DNA in the cell is modified. When the genes are changed, the proteins synthesized by the genes are modified. When the cell grows and develops, then, a plant with new characteristics from the different DNA is generated. Before genetic engineering, when humans selected the plants with certain characteristics or crossbred different strains, genes were manipulated. The genetic traits for modern corn were selected over time, starting with the early ancestor of corn, teosins, which is right here. This chart shows the basic steps of genetic engineering. This is an example of a single-cell bacteria. This is the simplest organism, and therefore the easiest genetically engineered. The bacteria has a ring called a plasma, in addition to their chromosome. Scientists can remove change and replace the plasmid. So they've taken it out here, taken a new gene, and they've inserted it into the plasmid. This plasmid with the inserted DNA is now called a vector. This is then reinserted back into the bacteria. Then when the cell reproduces, it does so with this new DNA insertion. This is a picture of transgenic rice. It has a resistance to a virus that would ordinarily kill the plant. Genetic engineering of transgenic, where an organism is created by the transfer of genes across species. Genetic engineering can also be used to do the same thing as crossbreeding, just more efficiently and faster. There are a number of reasons that genetic engineering can help us. They can make crops more resistant to diseases, tolerant of stresses such as salt, heat, or drought. They can develop soybeans that produce high yields of biofuel per acre. They can have a use of enzymes to create new drugs. They can develop vaccines to grow inedible products. There are many individuals that oppose genetically modified foods. Some of their arguments are the plants may then alter the weeds, creating another weed that would then be more resilient to the pesticides, herbicides, and other things we try to use to protect our crops. And in response to this, in the research that has been performed, the offspring has not been strong enough to survive. Some countries have put limitations on where the genetically modified plants can be harvested to reduce these risks. There's also concern about the long-term effects of genetically modified crops on the local ecosystem. What are the effects on insects, the birds that prey, or even our own bodies? The long-term research, once again, are not completed. However, the short-term research seems promising. It seems that as though the insects that are more effects are those that have adverse effect on that are affected more have the most adverse effects on the crops. The others seem to remain unharmed. There are many regulations to keep adverse effects to a minimum. However, there is still research being done in this area, and there is still much to be done. This is a rather new science, and we are far from having all the answers.